The second day of National Science Academy's lecture workshop on biotechnology, principles and practices held at Sri Shankara Arts and Science College. I can see a lot of familiar faces from yesterday's session and also some new faces today. A very warm welcome to all of you. Science is the artery through which the solutions of tomorrow flow. With this thought, today, straight away, we'll dive right into the technical session. Session 5. Before beginning our session 5, I would like to call upon Dr. S. Malati ma'am to introduce our speaker, Professor R. Puvanakrishnan sir.
A very good morning to one and all gathered here. It's my great pleasure for me to introduce our speaker of today's first session, Dr. Puvanakrishnan sir. He is an emeritus scientist, former director grade scientist, and head Department of Biotechnology, CSIR, Central Leather Research Institute, Chennai. He has completed his graduation in chemistry, post-graduation in biochemistry, and earned his doctoral degree in enzyme technology from University of Madras. He has obtained his postdoctoral fellowship from the University of Medicine and Dentistry of New Jersey, USA. Sir is a stalwart in the field of microbial protein and enzyme technology and has 10 national and international patents, 105 research articles in peer-reviewed journals, and 4 books to his credits. He is also serving as a reviewer and editorial member in several globally renowned journals, and he has guided 20 Ph.D. scholars. His rigorous research career is equally mirrored by his illustrious teaching contributions. He has taught leather technology for B.Tech and M.Tech students at Anna University for more than 25 years. He has been a recipient of Distinguished Scientist Award for Biological Sciences from Tamil Nadu State Council for Science and Technology. He has been our guiding pillar and source of inspiration. I am sure that the young minds who have gathered here would definitely get inspired by your speech, sir. Sir, on behalf of the organizing committee, I welcome you to share your experience with us, sir. I welcome you, sir.
Now I'd like to request Professor R. Puvanakrishnan sir to present the 5th session on enzyme technology applications in industry and medicine.
Good morning to you. We're happy to be here. This Academies, they have organized a lecture workshop. So before I start, of course, I have to wish Professor Muradhar, who is the architect for this lecture workshop, Professor Manuswamy, head of the department, Dr. Ramesh, and all my faculty here today. It's really immense pleasure to be here. I come here. I'm a regular customer here. The topic chosen is enzyme technology, its applications. So I will not be giving all details about intricate details. I won't go. I just like as Professor Muradhar yesterday did like a history. I would also follow your faithfully your steps. This will be a history of a leather industry how this biotechnology and how is it connected to leather industries. Or I call it as a leather biotechnology. You may not be knowing that leather biotechnology, if I call, you may be wondering, how is it that a new terminology is being created? I'll come to that. So in leather industry and research, we have few areas for enzyme technology, recombinant DNA, fermentation, and tissue culture. So different areas are there in CLRI. Not only that enzyme development, connective tissue biology research is there, in tissue culture and other models, animal development of animal models. We have environmental biotechnology. So like that, that a lot of biotechnology areas are there. Simply, we call it as a leather biotechnology. Something new terminology I'm giving to you. So before I go into the details a little bit, I would like to tell about my organization, where from I come. I'm from Central Leather Research Institute. It is from CSIR. CSIR, you should be all knowing. Council of Scientific and Industrial Research. We have 38 national laboratories. CLRI is one. You should be questioning me later. Why you should be thinking, why is it that CLRI is in Chennai? There is some reasoning. Because 70% of the Indian leather industry is in Tamil Nadu. And something, statistics, 70% of the Indian leather industry is in Tamil Nadu. It starts from Chennai. We have called Chromepet. You should be knowing Chromepet. The name itself suggests that the Chromepet and other adjoining area, we have a lot of tanneries are there. Tannery is the place where we tan the skin, is it not? Processing of skins is going on. Apart from Chennai, we have to go towards west. We are in Kanchipuram. You should be knowing that near Kanchipuram, about 35 kilometers, we start from Ranipet, We have Ranipet, Vellore, Ambur, Vaniyambadi, Paranambet, then little far, Erode. But come to the south, we have some tanneries in Trichy and Dindikal also. But beyond that, you don't see in Tamil Nadu any tannery or in the western side, eastern side also, you don't see any, coastal side, you don't see any tannery. Okay, this is the tanning industry in Tamil Nadu, as I told you. So how is it this leather biotechnology is important? You should be wondering. So it shows that 60% of the finished and leather goods are from Tamil Nadu exported. You know that the exchange value in terms of rupees, it's 40,000 crores per year. Last financial year, it was around 40,000 crores. So you should be wondering how much amount of the work is being done in the industry with regard to the finished skins, then footwear, then the leather goods. So it all amounts to 40,000 crores. So when we do like that, apart from that, when we say so much amount of money is generated, we also generate pollution also along with the money. You all agree. Once we generate money, the pollution comes along with that, especially in the parlor, that we call it leather belt. Leather belt, I mean, starts from Ranipet, then Vellore, Ambur, Vaniyambadi. So this is the leather belt. When you go by a train, you should be having a peculiar smell coming from Ranipet. I don't know. If you go by AC compartment, you may not be knowing. But if you go by general compartment, you should be smelling that nice smell. So what I mean, the effluents from tanneries polluting the Polar River basin. That is most important. Because originally the Polar was a really good one. So once the tannery, the effluents are discharged, even the color of the bed is changed. Who are the culprits for this calamity is that sodium sulfide and lime. Sodium sulfide and lime calcium oxide, they are the two irritants. They are the chemicals used in the pre-tanning processes. So when I say pre-tanning processes, you will be wondering, what are the other things? Because leather processing itself have three components. One is the pre-tanning. Number two is tanning. Number three is finishing. Okay. So the pre-tanning only, but the leather biotechnology or enzyme technology has a major part only in the pre-tanning processes, right? So I told you that the calamity is due to the sodium sulfide and lime. What they do actually in that, that the skin, before, we do the processing, before we processing, what they do, the previous day, they soak the skin in water. They remove all the salt. They remove all the salt, and then the next day morning, what they do, they make a paste with sodium sulfide and lime, and they apply on the back side or the flesh side there. They apply, and then they keep overnight. So what actually happens is the next day morning, next. So before I go there, I just wanted to show you that lime and sodium sulfide we use for dehairing, we call. Dehairing is the removal of hair. Okay. So that is the first unit process in the leather processing. So when we say lime and sodium we use for dehairing, and then the degreasing is another step that comes later. So the alternative technology when we call, instead of this lime and sodium sulfide, we use proteolytic enzymes. And instead of solvents and detergents, we use lipolytic enzyme. Next one. Well, there is major irritant from again the tannery is the solid waste. The solid waste come from different operations in the tannery. So first operation where we lime sludge and sodium, there's a waste. After we remove the hair, this is the waste going into the gutters. Instead of that, we suggest we use protease. The salt also is a major irritant. Again, this contributes to a lot of pollution load. The pollution load we call BOD and COD. Simply you call it a pollution. Instead of salt, we are advising to go for biocide. But a lot of biocides are there. The neem, mangosteen, cetyl trimethyl ammonium bromide. So a lot of biocides, alternative technology. What we do with this hair, chrome shavings, trimmings, these are the waste. So the waste, what we do, so we use the protease to degrade and then use it for profitably for other byproducts industry. So again, the flushings also, we use protease and lipase to degrade them. So this is only to show you that what are the solid waste generated in the tannery and what are the alternative technologies. Next one. So again, these they don't bother about the number. So these are the criteria for, that we have to adhere. The tanneries have to adhere. What are the total dissolved solids, biological oxygen demand, chemical oxygen demand, total suspended solids, chromium. So these are the parameters we have to adhere to. So there was, next one. Again, that there was a major problem in the leather industry about 20 years back. You know that Tamil Nadu, we have 100,000. That means 1 lakh tanneries are there, small, medium, and big. Suddenly, because there's a pollution control board, they brought a lot of restrictions. So all the tanneries were closed for some time, and all the workers were thrown out of job. The major problem is that use of lime and sodium. So the sodium, they said openly, sodium sulfide and lime you remove. Instead, you try to use enzymes. That's where the biotechnologist comes. Okay. So then we have shown actually that it can be profitably used. Enzymes can be profitably used in the pre-tanning process of leather manufacture. So the first step is the we call beam house process. I told you that in the leather processing, we have 3 units. One is the beam house process, where we remove unwanted components like hair, flesh, fat, everything. The second is the tanning process, where that we use the tanning materials to produce a stabilized fiber structure. I'm not going into the tanning part as well as the post-tanning process. Next one. So where is this enzyme technology where it comes in the actual leather processing? So it comes between the removal of skins and hides from storage, and the preparation for tanning. So in between, the enzyme technologist comes and he does the work. Next one. So these are the some of the operations where we use enzymes. One is we call soaking operation, where we use enzymes, all protease, mainly protease, some amount of amylase. The enzymatic deherring is a main process. Then we go for the baiting. This is also where we use enzyme, the protease enzyme. In addition, use lipase in another operation we call degreasing operation. Next one. Okay, this is only I was telling that. I was telling you that the previous evening we apply sodium sulfide and lime on the skin. Okay, so the next morning you take a blunt knife. A blunt knife is used to push all the hair. Along with the hair, we get the sodium sulfide, lime, everything, all the solids, everything comes, it goes into the effluent. So this is the major problem. Next one. So instead of applying, the sodium sulfide and lime, I told you how they are applying. The enzyme is applied like this. Now you can make a paste of this enzyme solution, and you can manually apply. This is one method of applying the enzyme. Next one. Instead of that applying manually, even you can also using the spray gun, you can spray the enzyme. It's another method of applying the enzyme. Mainly we apply protease for deherring. Deherring is removal of hair. So what are the formulations? What are the formulations we use in all the in other industry? One is we call it soaking aids, deherring formulation. We call it bait. That's the name given. Again, it's an operation where we use a protease. Then we have enzymatic degreasing where we use the lipases. Only to show you. Next. As I told you, I'm only going to give you a message. The message is that in the leather industry, what we are doing, we call enzyme technology or leather biotechnology. So what are the main enzymes which are being used? Is a protease, then lipase, then amylase. So these are the three enzymes we use in the pre-tanning processes or where the enzyme technology has got some application. Okay, next one. When you say enzymes, then you will ask that what are the sources of enzyme? Can you use any enzyme or any source can use? The question is that, so what are the animal sources for using the pig pancreas? The olden days, or even for bait preparation, we use pig pancreas, pancreas, even human pancreas also we contain a mixture of, you all know that in the 11th standard, 12th standard, you would have studied that human pancreas contains a mixture of protease, amylase, and lipase, is it not? So like that, the pig pancreas also, or it could be even bovine pancreas. So bovine means cow and buffalo, right? So cow and buffalo pancreas also we take that, we make a mixture, and we make a mixture and that preparation. So if necessary, even we can go for ammonium addition. So that is again the olden method. We can use plant enzymes. For example, the calotropis, called Tamil erkalai. So the erkalai, that latex also, it contains a powerful protease. Then java seed leaves, the java seed leaves, Rajasthan and Punjab region, we get it. Then ragi amylase, a pineapple proteinase. Then we have the adeno. In Africa, we have a fruit by name Adenopus brevis. These are the sources of plant enzymes. Next one. So nowadays, so we only go for microbial protease because that is where we can go in for large amounts. Again, the difficulty in collection of plant and animal sources are very difficult to collect the enzyme. So nowadays, all only microbial enzyme technology. So the enzyme technology, you slowly change into microbial enzyme technology. Then you will ask a question that how that can you go for any bacteria or any fungus anywhere? The question is that, can you think of using anything? It's not possible, right? Then you will ask a diseased, the diseased pathogen. Can you use for the as a source of enzyme? It's not possible, right? So what will the criteria is that it should be non-toxic. The bacteria or fungi you take should be non-toxic and non-pathogen. That is the criteria for choosing a bacteria or fungus. Next one. Well, you know that we say that we cry horse, and then we say that you remove this sodium sulfide, remove this lime. That is what the merit, one of the merits is that we get a better hair quality. What we mean is that when we use lime and sodium sulfide, the hair becomes a pulp, and then you can't use that for anything, any byproduct we can't use. But when you use enzyme, the whole hair you get as a byproduct, and then even you can go in for the production of amino acids or any other thing. Amino acids, definitely there is a unit in Pondicherry where they collect the hair from the leather industry and they make amino acids. So that is one of the major use of this enzymatic dehairing. And then you agree with me that there is less effluent problems in terms of BOD and COD. So when you use lime and sodium sulfide, the pollution load in terms of BOD and COD is very high. So when you use enzyme, have the less amount of BOD and COD. Again, you know that there are demerits also. When I say all merits, then somebody will ask me, do you think that there is no demerit when you use enzymes? You should think of that also. Only because the person who is an enzyme technologist will say, yes, it's very much advantageous. But there are some problems in a sense. Specificity of the person who uses the enzyme in the tannery, they should understand the properties of enzyme. For example, what's the pH in which we are using? What's the temperature? So this should be controlled. Then only it will be optimal application. And again, further opening we call, this is a leather technology one, where we use some lime and then for we call opening up of the fiber bundle. So this is now the message. So what I have been telling for the last 10-15 minutes is that the leather industry where the enzyme technology is used, the major enzyme which we use is a protease for dehairing operation, baiting operation, then soaking operation. Then there is going to be another operation later which comes even for the baiting operation also. You know that in addition to collagen, now I will just go in little into the basic structure of this skin. You know that we should be all knowing that you should have heard of the name collagen. The collagen is the fiber bundle that is left out when we do all this enzyme technology or treatment. You don't touch the collagen, because once you touch the collagen, the structure goes out, and then you can't use the leather. So the collagen should be retained. It should not be touched. Then what else you do with this enzyme is that there is a reticulin, elastin. There are so many unwanted proteins in the skin. So those unwanted proteins only be removed by this operation, baiting. The globulin, reticulin, elastin, even keratin. So be used by this baiting operation. And the skin becomes very smooth. Only collagen only will be remained. Right? Next one. Well, you may be asking another question. Somebody might ask, you are telling so many things. Have you developed a protease in CLRI? Is it not? You should be next question. Am I right? So we have also developed a protease. So by name, bacteria, we have developed a bacterial protease from Bacillus, and we have shown that it has got good activity and that it works. These are the characteristics for the enzyme which you don't bother. The message is that CLRI, we have produced a protease and it's given to the industry, we have marketed it. In addition to that, we call a cocktail of proteases. A fungal protease also has been developed. Again, it's given to the industry. So like that, once the industry man comes, you show the shelf where I have 4 or 5 products of enzyme, he can choose one. That's the way, is it not? When you go to the shop also, there will be 5 different, that brands. You choose the best one. Next one. So this, I told you that conventional process use the lime and sodium sulfide. So when we use enzyme, there is no necessity for the use of sodium sulfide. This is the most, the criminal is sodium sulfide. Because of this only, we have a lot of problems. Use less amount of the lime also. There are so many economical benefits, environmental benefits are there, you don't bother. The message is clear that we use protease for the dehairing, weighting, and soaking operation, right? Next one. So this one is only to show you the structure of skin. You should be all, these are, you would have done in 11th and 12th. You take the skin, we have the epidermis, then we have the hypodermis, we have the dermis in. So some type of skins. So this is the, just to show the structure of a skin. The epidermis, dermis, hypodermis. You see the structure of certain skins. For example, ox one or New Zealand sheep or Australian sheep, there the fat content is more than very high, 20 to 30% fat content. While our Indian, that skins, the sheep skins or that our buffalo or cow, we don't have that much amount of the fat. So you will ask then what? If you take all the sheep skins or New Zealand sheep skins, then Australian sheep skins, and go for tanning without proper treatment, the final, the product is not good because of the oil. There are a lot of patches will be there, so you can't sell those products. So what we do in that case, then how to remove the fat before you go in for processing? There's a question. Can you remove the fat? The presence of excess fat creates this type of problems. Now, these are the leather technology wordings. The simply to put it in a simple way, if you don't remove the fat, it ends up in lot of problems. The final, that leather is not good one, right? So how to remove the fat? Next one. There are many methods. One is that we call solvent degreasing, where solvents like kerosene, trichloroethylene, petrol. So these are the solvents normally we use for the removal of fat. So the major disadvantages are that they're toxic, hazardous problems. The workers are subjected to all these, solvents. The number two, another method is there where we call it aqueous emulsification. That means use detergents to remove the fat. Lot of detergents, anionic detergents are there, cationic detergents are there. So we get those things and then we use to remove the fat. There is again yet another method. Next one, please. Then again, the enzyme technologist comes here. Enzymatic degreasing. There use lipases for using removing the fat. So what are the advantages? That we don't use solvents, don't use surfactants. And again, when we go in for this enzymatic degreasing, that means you take lipase and treat these skins having a lot of fat. So you get a byproduct. What is a byproduct? You just all doing biochemistry. So when you treat this lipase with a fat, what will be the final product? I'll tell you the answer, but simple answer. Take a lipase, treat with your oil. What is you put glycerol and fatty acid. Simple, isn't it? You get glycerol and fatty acid when you treat the oil with lipase. What is the importance? So what you do with the fatty acid is that the fatty acid is taken for soap industry, right? So it's only what is a soap? Then the next question, what will be a soap? It's only a sodium or potassium salt of fatty acid. So the point is that here the end product, when we use lipase, we get again valuable byproduct by name fatty acid. You can use it for any industry. Next one. Again, you come to the source. How this lipase, what are the sources of this lipase? One is that, again, plant lipase from castor bean, one good source. Pancreatic lipase. Pancreas got a source of protease, lipase, and amylase, right? Our microbial lipase could be alkaline or acid or neutral. Next one. That there are the message. What is the message till now is that you can use protease, you can use lipase, and what are the best sources? Microbial sources. Why we call it microbial enzyme technology. So now you will be asking. You should have read another important aspect by name immobilized enzyme. That not you should have read immobilized. Have you read or is it not? Immobilized enzyme. Immobilized enzyme is nothing but take a soluble enzyme coupled with a substrate. It looks like insoluble. That substrate can be inorganic or organic, anything it can. There are many methods are there. You couple the enzyme with the support. It looks insoluble, but the activity is retained. That is the advantage of immobilized enzyme. What are the other advantages? Say that enzyme is there, you dissolve in water, you simply use it. Why you think of all this immobilized is that the major advantage of using immobilized enzyme is reuse the enzyme. Before I go there, so enzyme is native and another is a modified. Modified we mean immobilized. There are many methods are there. Entrapped enzyme or bound enzyme. Could be matrix entrapped, micro encapsulation. Could be by adsorption. There are many methods are there for doing immobilized enzyme. Okay, the next one. What are the immobilized enzymes? What are the disadvantages of the soluble enzyme is that soluble enzyme could be used only once. Once it is used, it's again thrown out or thrown out into the gutters. Again, it goes as a pollution waste. One is the cost of enzyme. Instead of doing one time, using enzyme one time, if I can use 10 times, so the industry feels that they're very happy, they can use the enzyme repeated. Again, large reactors with high capital cost, major advantage of enzymes. To eliminate all these disadvantages, immobilized enzyme. Next one, please. Lot of advantages I told you that immobilized enzymes permit reuse, and it could be operational. It could be any method, batch or rapid. So many methods are there to produce by fermentation. And the major advantage of immobilized enzyme is that you should have heard of the optimal pH of an enzyme. Is it not? If you take trypsin, the optimal pH is 8.0. You all know that. Or pepsin, take only 2.5 or 3. Optimal pH. So by immobilized enzyme making or preparation, you can change the optimal pH. That's a major advantage of using immobilized enzyme. Have increased storage and temperature stability. So these are some of the properties. You can question me. There are so many other important properties. Next one. Well, there is another area. Soluble enzyme is one. Using immobilized enzyme, number 2. Immobilized whole cells. There is another method by which, instead of using support, take the whole cells as such. It could be bacterial or it could be fungal whole cells. Again, entrap into a matrix. Entrap into a matrix. Then that matrix entrapped enzyme could be used in the industry. We have actually in CLRI, we have developed an immobilized enzyme product for use in the leather industry. And the support, you should be knowing, it's only a river sand we have. River sand we have used for immobilizing enzyme from pancreas. We have prepared the pancreatic extract and then immobilized on sand. Then we have shown that immobilized enzyme product could be leather industry. But there are certain disadvantages. I think it comes in masters, if I'm correct, immobilized enzyme. Masters comes as a chapter, all knowing that. So what are the, again, advantages and disadvantages? I told you that. Because the immobilized enzyme, there are many supports are there. In addition to, we have used, I said sand, there are glass, there are so many plastic, nylon, there are so many supports are there where you immobilize and reuse. Sand is the cheapest. But again, the problem is that the loading capacity for sand is low. That's why we couldn't use that product. We couldn't use it very effectively. There are so many other supports which we can use. Next, this is the immobilized whole cells. I told you again, this is better than immobilized enzyme. Matrices are alginate, agar, polyacrylamide, and carrageenan. Some of the matrices. So only tell you that this is there are methods for using enzymes. Instead of native enzyme, you can prepare an immobilized enzyme or immobilized whole cell product. Right? Next one. Well, I told you about the using by this enzyme for dehairing and then removal of hair or removal of oil or fat. We call it degreasing. There is another process by name curing, where you use lot of salt. We call it curing process. Sodium chloride. The salt is very much used. Again, the salt is a major pollutant. To remove from the effluent, removal of salt is a major problem. So what are the alternatives for removal of the salt? Is that using a biocide instead of either chemical bactericide or fungicides you can use. The alternative will be use of biocide. The message is that for curing process, we can use biocides. Right? So what are the biocides? Next one. Lot of biocides are available. We have the neem oil, garlic, cetyl. This is also biodegradable chemical. Then we can use the puvarsam, that the source we can prepare. Then Korai puli also they have shown. So like that, there are so many alternatives for preparing this biocide. Next one. Well, so this is the message with regard to the leather industry and the enzyme technology, right? Now, there are many other industrial applications for enzymes. I told you that leather biotechnology, I'm not touching all the applications because I am from the leather industry. I thought that so you should be all knowing that it's only 30 kilometers leather industry, tannery. Maybe I would request our department, Dr. Ramesh, to take you to a leather tannery where you should have a feel of how they are doing. You know that the tanning is the age-old industry, and then once you enter, get some smell you get, and then you should be seeing that how they do the manufacture the leather, whether they are using enzymes, what are the methods for treatment, how they are using alternative technologies. So that maybe we will try to maybe request our head of the department, take you to a good tannery and show. Okay, there are many other industrial applications, as I told you. Even the detergent industry, there is a lot of applications of enzymes. So you should all know that, is it not? In the TV, you see that the mother-in-law and daughter-in-law, they will be fighting. Is it not? You like it. The mother-in-law will use 200 grams of this detergent, and then you see, well, I'm using so much detergent. But the daughter-in-law is very smart. You say, no, why you use 200 grams? Only 1 gram or 2 grams. Finally, the daughter-in-law only should win, is it not? You all belong to the daughter-in-law side. So why this that 1 gram or 2 gram? How they work? Why the 200 gram? Why it doesn't work much is that it contains enzymes. I'm not giving all details about other applications. What are the enzymes which are used in the detergents? The next question will be, what are the enzymes? One is a protease, amylase, kinase, lipase, cellulase, manganese. These are some of the enzymes which are used in the detergent industry. Because the detergent industry also, you know that it goes for about 100 billion or 200 billion dollars. Everybody is using detergent, whether it's a chemical detergent or a enzyme detergent. Okay, so the industrial application, dairy industry, even the textile industry for desizing, they are using enzymes. Lot of applications. I'm not touching all the applications. Instead, I thought that, well, before I go for the medical applications, we have brought out a book. You know, the title is Microbes and Enzymes. It's a leather biotechnology. We have made a book and then given to your college. You know, Sankara College, I have given a copy also. Kindly go through that book and then you will know that how these enzymes are being produced and then used, how they are better, a comparative analysis also. Coming back to the medical applications, you may ask why industry only enzymes you don't use medicine? There are again lot of applications. Even medical use also we show that enzymes how they are used in the diagnostics. When you go to the physician, they are immediately they so many enzymes, measure this one, that one. Again, in the as and treatment also, enzyme treatment also, it's let us go through one by one. So as a marker is a diagnostic marker in clinical medicine, and another is a therapeutic or medical application. I'm only give general only. I'm not going to the detail. Next one. We have the global enzyme market size. Even in dollar, you can convert to rupee, 1 billion. Okay, can convert from billion to dollars. In 2024, that US dollar 17.5 billion. Okay, so this is the estimate. First estimate. The first one I would like to show you cardiac marker. What are these cardiac markers? Suddenly you go to have chest pain and you rush to the doctor. And we can't. Got lots. The doctor immediately writes creatine kinase MB. That's one important enzyme. You all know that, isn't it? Creatine kinase MB. He wants to measure the creatine kinase. Another is a LDH, lactate dehydrogenase. These are some of the apart from that, lot of protein markers are there. I'm not touching it. For example, troponin. Troponin is another important, but it's a protein. I'm not touching. I'm only restricting myself to the creatine kinase is a dimeric molecule. There are two forms, the two subunit forms of CKMM and CKMB and CKBB. Okay, so out of this CKMB, very specific for cardiac as a cardiac marker. During muscle damage, release muscle and then detectable in blood. Okay, so the CKMB activity is to measure. What is the normal value of the CKMB is less than 20 international units per ml. We don't bother about the units, only know that CKMB is an important cardiac marker used in medicine. Originally, they were using only CK, creatine kinase, only they were using, but nowadays they are using CKMB, the subunit. Next one. Again, we see lactate dehydrogenase. The lactate dehydrogenase, again, composed of two subunits. There are 5, if you go by electrophoresis, go by this, any electrophoresis, even by paper or any other form, you can see 5 that we can see that some level of LDH1 indicates that heart muscle is damaged. So LDH1 is very important, or even you can total LDH also you can measure. So you just try to understand that creatine kinase MB and LDH are two important enzymes which are measured as markers for cardiac problems. Next one. Then again we have a liver marker. I am not giving all the enzymes, only the enzymes which are used presently, which you come across very easily, very much. Another is one liver marker, as we call it alkaline phosphatase. Okay, the alkaline phosphatase again it's a hydrolase. So this increases due to liver disorders such as hepatitis, liver cancer, cirrhosis. So this alkaline phosphatase normal value is between 220 to 140. So it's very important enzyme when we go in for the liver condition. For example, it could be jaundice, or it could be liver disorder, hepatitis, liver cancer also gets enhanced. Next one. Pancreatic marker. When we say pancreatic marker, what is the pancreatic marker? Is an amylase. So the amylase gets increased in the pancreatic problems, especially pancreatitis or pancreatic tumor. This amylase gets increased, and then again it could be easily estimated. In the laboratory also, amylase or protease or even CKMB also, there are kits are available. Right? Next one, please. So we have a bone marker. Bone marker we call tartrate resistant acid phosphatase. We call it TRAP. Put it in a simple way, we call it a TRAP. So it's a glycosylated metalloenzyme Okay, it's a marker for bone resorption. Again, in arthritis, rheumatoid arthritis, osteoporosis, metabolic bone disorder. So these are some of the disorders. So this particular enzyme is very easily can be measured. I'm only giving very, very important one, which you come across. Next one. Then we have a renal marker. Renal means kidney disease. Call it renal marker. It could be any kidney disease. For example, this is for early detection of diabetic retinopathy. People who have got diabetes, they end up with kidney problems. So the diabetic retinopathy, so it's NAG is measured. NAG is N-acetyl beta D glucosaminidase. Okay. So this measurement of urinary NAG for screening diabetic renal dysfunction. Next one. If I want all details also, we can give you. We have published a book on, there's enzymes, how they are used in diagnostics, how they used in application. That means, yes, treatment as a drug. So we have got the book. Again, we have given the book to our Kanchi Shankara College. You can just go through it and then come out with questions. The next one is, well, I told you some of the markers, enzymes which are used as markers for diagnosis. Now, do you ask whether enzymes can they be used as therapeutic agents? Can they be used as a therapeutic agent? Therapeutic agents means what? You can use orally, or it could be used by intravenously, anything. You all come with, because we have a stomach disorder problems, pancreatic disorders. So when especially indigestion problem, I am getting gas, or it's not food is not getting digested. Go to the physician. The physician simply gives you a pancreatin. Is a pancreatic mixture only. Only thing is, it is required at a very high degree of purity. Pancreatic enzyme, you prepare at a very high degree, and then the preparations are available as lyophilized pure preparations with biocompatible buffering salts. Because in the industry, when we use the pancreatic mixture, it need not be very highly pure. It could be simply an ammonium salt mixed, which can be used. But when we go in for the medicine, so it should be very highly, thoroughly purified, then only it could. So for parenteral administration, they should be exhaustively, parenteral means by injections. When you administer, it should be exhaustively purified to eliminate toxic. Next one. So again, the insufficiency leads to indigestion. As I was telling you that pancreatic enzymes, the insufficiency leads to indigestion. The pancreatin is the drug that from pancreas. It contains protease, lipase, and amylase. Okay, so bovine pancreatic enzymes are better source for preparation. Next one. So this is urokinase. You should have heard that. Again, when a person gets heart attack, initially I told you when you get heart attack, as diagnostic, this is being used. CKMB, or that LDH, or as a protein, troponin. They are used for diagnostic markers, but this is for administration. Again, this is administered intravenously, urokinase. This is an important one. Urokinase are a plasminogen activate, serine protease, treat acute myocardial infarction, the thrombolytic drug. Okay. So because the thrombus should be degraded, because that's why we call thrombolytic drug. Therapeutically important fibrinolytic enzyme we call. Again, we can give so many names. So this is again another important for the enzyme applications as a drug. Okay, next one. Again, this is one of the streptokinase. This is also for cardiac problems. When it has been proved that this is better than urokinase. For 30 years back, they were using urokinase. Nowadays, they are using streptokinase. Again, converts, how it functions is converts plasminogen to plasmin. Right? The chemical, how the enzyme acts. Okay, so it's again, we have shown that it's a thrombolytic agent. Intravenously, it's given immediately after the onset of a heart attack. Immediately when you rush the patient, within 1 hour, if you can rush a patient to the clinic with a heart problem, they are able to save the patient by administering urokinase or streptokinase. Nowadays, again, modern times, there is another drug which is enzyme drug, which is next one, please. So again, this one is a tissue plasminogen activator. Call it TP. When more than one heart attack occurs, drug can be. So this is even better than urokinase and streptokinase. You'll be asking why. These enzymes are better. This particular TPA is better. Is that it's not antigenic. It doesn't cause immunogenic problems because when you use these enzymes intravenously, then you will ask a question. Can they not produce allergic reactions? Are they safe? That's the first question you should ask when you administer a foreign protein into the living system. That is why you have to ask the question, and this plasminogen safe, not antigenic, don't cause immunogenic reactions. Next one. One is asparaginase. You have also heard of this asparaginase. Converts asparagine to aspartic acid and ammonia. Where we use this asparaginase is that in cancer. The cancer medicine, asparaginase. How it works? Cells require a high amount of asparagine proliferation. So once you get a cancer, the leukemic cells produce a lot of asparagine. So to stop that particular process, asparaginase cells in depletion, better is best to use lymphocytic leukemia. For that, it's being used. Asparaginase alone or in combination with L-glutaminase is the best one. So we can use it alone or in combination with L-glutaminase is another enzyme. Also, you should have heard of that. With stomach problems, I have that some more acid, that we irritation in the stomach, or even in the surgeries, they prescribe the enzyme, call it serratiopeptidase. Hopefully, you have heard of this serratiopeptidase. So, what is it? Serratiopeptidase. It's a proteolytic enzyme. Molecular weight is about 50,000. So, it contains a zinc. It's all chemistry. Don't bother. This is an alternative to steroids. Peptidase is an alternative to steroids. So it's prescribed in various conditions, surgery, orthopedic, gynecology. So it's equivalent, diclofenac is equivalent to diclofenac. You should have heard of diclofenac, is it not? In all over India, the is an anti-inflammatory drug, very well used by so many people. So even available in the market, you go and then say, I want a diclofenac. Immediately, it's an anti-inflammatory. Serratiopeptidase is equivalent to diclofenac. Again, is a collagenase. I was mentioning about this collagen. When in the earlier also I was mentioning the word collagen. In the leather industry, you are not supposed to touch the collagen. That is, in the leather processing, the collagen should not be touched. But here, in the applications, as a drug when you give, the collagen is getting degraded by using a collagenase. Collagenases are used in medical, pharmaceutical, food, cosmetics, and textile segments, in burns and ulcer treatment. Okay, the collagen is a matrix metalloproteinase, helps to break up and remove dead skin and tissues, help in repair mechanism. This is the way the collagenase acts. The difference I told you. You retain collagen in the leather processing, but you break down this collagen in the application when you put it as a drug in the human system. Next one. So this is another one that we call it hyaluronidase. Also, you should have heard of this hyaluronidase. It catalyzes the hyaluronic acid. So they are using in ophthalmology as an adjunct to local anesthesia. How it's being used? Hyaluronidase. Therapeutically, in combination with other drugs to improve better penetration of chemotherapeutic drugs in tumor also they are using. So hyaluronidase, see, this is the message. Yeah, okay. So stop. What I would do, these are my students responsible. My picture was taken about 10 years. These are my students, they're all doing very well, both in India and abroad. So they are responsible. Only part of my research group only it's shown here. Okay, so it's a big group we have. So I have to thank them. So whenever I start preparing a lecture, finally I thank my students. Because of them only, I'm here to speak with you. So
Thank you,
I
sir.
would.
The session is now open for interaction. Are there any doubts?
Sir, you told about creatine kinase. What does MB stands for, sir?
No, it is a subunit we call, that they have given some, that letters. One is MB, another is LL, like that. They have given, there is no, I don't think there is any specific meaning for MB. Differentiate the subunit. I don't think there is any. But get him.
Very excellent lecture, sir. Our comprehensive account on leather industry and use alternative biotechnology to leather industry. Regarding the proteases, how cost effective upscaling and commercial levels, sir?
Our Professor Manusamy asked a very pertinent question. This is what even from the leather industry we are being asked. So whenever I say that enzyme is effective, the industry compares immediately the cost of 1 kilo of the sodium sulfide, 1 kilo of lime. What is the cost? What is the cost of your enzyme? Can you make an enzyme for 20 rupees, 1 kilo? Not possible. Then we say, you look at you remove all the sodium sulfide or lime, environmentally pollution you avoid. The cost you are spending for, I'm coming to the scale up later. The cost, that you are spending for the production is getting equalized. For example, when you treat the effluent, you have to pay a lot of money to the effluent treatment plant for treating the effluent containing the sodium sulfide and lime. So when you use enzyme, that problem is not there. Not only that, even the leather which is made by using enzyme, it's more better when compared when it's used by sodium sulfide and lime. But the industry, they don't agree. You give me whatever the cost, 20 rupees means give the enzyme for 20 rupees. But maybe it's possible later when you all go in for recombinant DNA technology. This is, that is conventional technology, using that getting enzymes, add ammonium sulfate, prepare, and then mix up. It could be bacterial, then use it in a partially purified form. We are not going for final pure. That means very highly pure enzyme we are not making for leather industry. It's only partially purified. One point. But when our professor was asking, the scale up, that it is possible. Only when we go for a scale up, only we can use. It could be it could be by, that submerged culture fermentation, or it could be a solid state ferment. We have done both. So it depends. For example, bacteria, when you go for even if higher, scale up, first you make a scale up initially in the flask, then you go for 1 liter, then you go for a fermenter. You have also a fermenter here, 4-liter fermenter, 500-liter fermenter, 1000-liter fermenter. So a bacterial fermentation is by using a fermenter. Instead, you can go for a fungal also. When you go for a fungal, this type of very strict, here, the most important contamination problem when using a bacteria and produce an enzyme. The contamination problem is a major one. But when you go for a fungal, you go by trace. There are many methods. I'm not touching all those points. So you go by trace. The trace, you have your bed where you have a solid substrate. It could be wheat bran or any other one. There you inoculate the fungus, and then there's another. So both the methods we can prepare.
Any other questions?
Sir, why can't we use recombinant DNA to produce enzymes?
Pakat, please come.
Sir, why can't we use recombinant DNA to produce enzyme instead of using pig pancreas, sir?
Recombinant DNA?
Can we use recombinant DNA to produce enzymes, sir, instead of pig pancreas?
Very difficult. That recombinant DNA technology, normally, that you have the protein, finally purified protein. Normally, they go from the microbes. Normally, microbes is the ideal way. A bacteria or fungal. Finally, first you extract the protein from the bacteria, and purify. Then after purification, you use your recombinant DNA tech. But it's not done from the pancreas. It's also possible. Your question, I also say yes. Then you very highly purify. That means a very highly homogeneous preparation, wherein you do by electrophoresis, get only one band. That's one protein going for.
Okay, sir.
Otherwise, normally then nowadays all microbes only.
Sir, after the enzyme treatment, where it is released, sir?
Then treatment.
Yes, sir. For
Effluent.
dehairing process.
What?
For dehairing process, no, sir. We are using enzyme, no, sir. After that, where it is released? Yes, sir. Where it is released, I am asking. Sir. After treatment.
Oh, okay. For talking about cannery, they have a different way of canning. That canning, post canning, all the that things are. Your question is more pertinent in a sense, important in a sense. If you go to a big tannery, they have even effluent treatment plant, very big tanneries. But you may say that in Ambur, there are about 100 small tanneries. The question is they don't have a treatment. What they do actually, there is another terminology we call common effluent treatment. They call the CETP. You should have heard in Ambur, Vaniambadi, Vellore. CETP, how they do all these small tanneries, they have the channelized, they put a big tube and they take to a particular point, and they put the effluent like that. The effluent. A common effluent plant is set up, treat those. There are many methods.
Okay, sir. Sir, you told immobilized enzyme can be used, reused, no, sir.
Yeah.
How many times it can be reused?
Okay. It is again another important question. For example, I told you about the way sand, it was my PhD problem, where I used the river sand for immobilizing and the cheapest support. You agree with me? Not very high, expensive, very cheapest. But there is an inherent disadvantages. What is the disadvantage? So you know that it doesn't have a loading capacity. The amount of protein loading capacity for that carrier very much less. That's why, but there are many other advantages. But what's your specific question?
How many times it
Okay,
can be
how many
reused?
times? I have used for 3 to 4 times my immobilized product. For example, if you use that glass, glass, small, very fine particles or nylon, or there are so many, lot of, the plastic, the new plastics they had for immobilizing it, and they're using 100 times. Instead of using a batch process, put it in a column, immobilized enzyme, you put it in a column, and then you pump the substrate. When you are doing like that, it could be even 50 times, even 100 times. Depends upon the greatness of your immobilized enzyme.
But the effectiveness remains the same, sir.
Effectiveness means what you want. Effectiveness.
The enzyme.
Yes, effectiveness should be the same. But again, to offset the loss, say that some amount 2% or 3% or 5% loss. So you recuperate, add another, after one use, add another 5% of the enzyme, so that to compensate the loss.
Okay, sir. Thank you, sir.
Next, I would like to call B. Vaishnavi of 3rd BSc to end section 5 with vote of thanks.
கேளுங்க அடிச்சு தள்ளி. Any other question கேளுங்க.
Any other questions?
No question. I think okay, வாங்க குயிக்கா வாங்க, இங்க வாங்க, பக்கத்துல வாங்க, because the next speaker is ready, so I have to. So I'm a local man, you can talk with me also later. Quickly.
Sir, any catalyst is used in leather processing?
Any
Catalyst is used in leather processing.
carrier.
Catalyst.
Catalyst. Catalyst or catalyst? Catalyst.
Catalyst.
Same is a catalyst. Itself is a catalyst. Chemical catalyst, and biological catalyst. We are using a biological. That is the catalyst. So, thank you very much. That for your kind patient hearing. I'm here only. You can come out with questions. Thanks.
Good morning, everyone. I extend a really hearty vote of thanks to our chief guest, Professor R. Puvanakrishnan sir, who spared time from his busiest schedule to grace this workshop. Today, we had a wonderful opportunity to hear your thoughts on enzyme technology applications in industry and medicine. The session 5 began with the brief introduction of leather industry and research, the importance of leather biotechnology, how sodium sulfate and lime are responsible for dehairing and a lot of pollution, how enzymes can be an effective alternative for this. He then gave his remarkable insights on enzymatic dehairing, applications of enzymes by paint and spraying technique, how protease, amylase, and lipases are used predominantly, what are the sources of enzyme, that is animals, that is as in pigs, plants, latex leaves, pineapple, etc. He also shed light on the merits and demerits of enzymatic dehairing. He then also told us how bacterial proteases are used. Lastly, he told the applications of enzymes in various different areas, as in medical and surfactants. He ended the session with how enzymes are used as therapeutic agents in pancreatic enzymes, urokinases, streptokinesis, tissue plasminogen activators, asparginase, streptopeptidase, collagenase, and hyaluronidase in chemotherapy. Thank you very much, sir. Next, we'll proceed to session 6. Yesterday, we had a memorable experience of listening to Professor P.B. Seshagiri sir, who presented lecture on stem cell biology and technology practices. Now, I'd like to request him to present the 6th session on stem cell biology and technology applications.
Sir, are we audible, sir? Can you hear us, sir? Sir, are we audible, sir? Sir? Sir, we couldn't hear you, sir.
Hello? Hello? Yeah, I can't hear you. I can't hear any audio from your side. Any
Now,
reason?
sir, now could you hear us, sir? Sir, now is it audible?
Hello, can you hear? Can you hear?
My voice also. So now you please check. Now you please check audio audio, sir.
Hello, can you hear?
Are we audible, sir?
Yes, yes, now I can hear. All right, just give me a few seconds. I will share my slides. Can you see my slides? Okay, I will start this second lecture today. Thank you again to the college and the organizers for providing this opportunity. Yesterday, I described about the basics of stem cells, stem cell biology, how these are derived, and what are the properties, and so on and so forth. So basically, I covered on the biology of stem cells and as well as a little bit on the biotechnology of stem cells as well. So today, what I'll do is actually give an overview of why are we into stem cell research and stem cell technology, and what are the potential applications or benefits that the stem cells offer. That would be basically the kind of the profiling of my talk today, and so I'll just go with what I did yesterday, the genesis of stem cells and types, which we already knew quite a bit yesterday. And importantly, I was stressing on the stem cell differentiation biology and the biotechnological implications. So I'm just going to touch a little bit on that and then go forward with the potential applications that the stem cell research and stem cell technology actually offer, and what kind of future prospect that one can expect. This is a recap of the slide that I showed yesterday to recapitulate about what are known as the pluripotent stem cells. And so if they are derived from blastocysts from the inner cell mass, we call that as embryonic stem cells. It can be from any organism. And way back in 1981, it was derived from the mouse for the first time in the world by Martin Evans' group, and later from the non-human primates by Jamie Thompson's group, and again from Jamie Thompson's group, the human embryonic stem cells were derived in 1998. And so these were the stories, historically speaking, about the derivation, rather establishment of embryonic stem cell lines, which I described in detail yesterday. And the moment the first human ES cell lines were derived in 1998, the whole world took by storm, looking at the enormous potential that the stem cells could offer. You know, the stem cells are mother of embryonic stem cells, particularly, are the mother of all stem cells. And therefore, it's very clear from the basic biology point of view that the ES cells can produce virtually every type of the cell that our body contain or any mammalian organism contain. And therefore, it was very exciting those days to see that, hey, here is the method where you can use stem cells as an approach or an alternative to drug treatment to a number of human diseases or disorders. Because there are a number of disorders and diseases, for example, genetic disorders, so on and so forth. I will not go into detail, but they cannot be simply cured by administering drugs, by oral administration of drug or infusion to the bloodstream or transplantation into a particular target site. And these are very challenging and still a very limited medical therapies that are available. And therefore, an alternative was a stem cell based approach for treatment of a number of human diseases and disorders. But at this time, if one asks, is there a stem cell vial that is ready to go for treating a disease, the answer could be yes and no. I will tell you that a bit later. But at that time, when the ES cells were derived, these were, there are two concerns that one need to remember in 1998. One is these were derived by scientists in a research laboratory. So those cell lines that are produced in the research laboratory conditions are good for academic research purpose, to do research, and maybe to some extent R&D kind of an approach. But they are completely unfit for use in the human, because you need to have a grade, a clinical grade, which is safe and which is efficacious. So the safety and efficacy are the two cardinal criteria for any cell-based material to be injected to humans. And therefore, what is known as a clinical grade embryonic stem cells or any other stem cells is what is required if it is to be tested in the human. Because there needs to be two things that need to be determined unequivocally. One is safety of the cells after infusion and efficacy of the cells. It should do its function. And therefore, all the cell lines post 1998 that were available were all not clinical grade, they are research grade. That was one major issue. The second issue is that the embryonic stem cell derived, let's say, from a donor A, even if it is a clinical grade, let us say, will not be compatible to the recipient human because of the haplotype mismatch. It's like organ transplantation. If there is a mismatch, the organ will get rejected. Similarly, if the cells are infused and if there is a mismatch, those cells will get rejected and there could be an immune reaction. And therefore, you need a histocompatible, haplotype compatible, haplotype matched ESLs. And that was the challenge those days. So that challenge was overcome initially by Shinya Yamanaka, and there's a picture here. And that person did a fantastic basic biology. What he did was to take any cell of a donor, let's say a patient here, and you take any cell, for example, fibroblast from the skin or buccal cavity or whatever, and forcibly induce 4 transcription factor cocktail. There are a number of cocktail transcription factors that were described. I will not go. It's OCT4, SOX2, KLF2, MIC1, MIC-C or Lin28. So these are the combination of 4 transcription factors. If they are introduced to any somatic cell, that somatic cell will convert itself to embryonic stem cell-like cell. So what in effect we have is any somatic cell which is induced to become embryonic stem cell-like cell or a pluripotent stem cell. So these cells are therefore called induced pluripotent stem cells. they are pretty much 99% identical to the embryonic stem cells. So you have two types of pluripotent stem cells, like I mentioned yesterday. One derived from ESL, embryos, embryonic stem cell. One derived induced rather from a somatic cell, and these are called induced pluripotent stem cells. So this gave us an opportunity that here are a methodology where